At the exact intersection of a patient's immune system and a tumor cell lies a microscopic synaptic cleft. This is the battleground where cancer cells deploy proteins to shut down the body's natural immune response. For patients with triple-negative breast cancer, or TNBC, survival relies heavily on immune checkpoint blockade therapies. These drugs are engineered to target this exact synapse and physically block the tumor's suppressive signals. To determine if a patient is eligible for these checkpoint inhibitors, doctors use a standard immunohistochemistry test, or IHC, to detect the presence of a specific target protein called PD-L1 on the surface of the tumor. But clinical data reveals a paradox. A significant subset of patients who test entirely negative for the PD-L1 protein still show significant clinical improvement when administered the targeted drugs. Look at identical tissue samples from the same tumor. The left shows the standard test yielding a near-zero detection rate. The right shows dense positive markers for the exact same target protein, achieved by adding one preparatory step. The standard diagnostic pipeline contains a mechanical flaw. A negative stain result does not always indicate that the tumor lacks the targetable PD-L1 protein. To solve this diagnostic failure, a coalition of researchers initiated a multi-center real-world study originating from hospitals across Taiwan. This flowchart outlines the scale of their investigation. The team collected archived pre-treatment tumor samples from 154 TNBC patients across varying stages of therapeutic management. They established a baseline by testing these samples using the FDA-approved DACO-288 monoclonal antibody, which serves as the gold standard tool for clinical PD-L1 detection. The researchers operated on a specific hypothesis. The PD-L1 protein is physically present on the cell membrane, but the diagnostic antibody is blocked from seeing it by post-translational modifications. Specifically, they pointed to N-glycosylation. This is a normal biological process where cells attach complex, branching sugar molecules to their surface proteins. Before tracking how this affects patient outcomes, we have to look at the molecular architecture of the protein itself to understand exactly how these attached sugars break our primary diagnostic tool. This schematic maps the PD-L1 transmembrane protein. The gray rectangular segments represent the extracellular domains extending outward. Notice the 4 red markers along these domains: amino acid residues N35, 192, 200, and 219, where N-linked glycosylation physically attaches. Cancer cells protect the receptor by coating these 4 specific sites in thick, branching sugar chains. The standard 28-8 diagnostic antibody is engineered to bind here, but when applied, it physically collides with these bulky sugar branches. In biochemistry, this physical obstruction is known as steric hindrance. The sheer volume and density of the sugar chains create an impenetrable shield around the binding site. The clinical assay fails because the testing antibody literally bounces off the tumor's carbohydrate armor, leaving the underlying target undetected. If complex sugars are the physical barrier blocking detection, the logical intervention is to strip those sugars away before applying the diagnostic stain. To execute this, the researchers applied PNGase-F, a highly specific enzyme designed to cleanly cleave N-linked oligosaccharides from glycoproteins. Washing the tissue sections with this enzyme dissolves the steric hindrance, unmasking the true density of the PD-L1 receptors on the tumor surface.
Because PNGase-F only targets the carbohydrate attachments, the underlying amino acid structure of the PD-L1 protein remains intact, leaving the exact target epitope preserved for the antibody to recognize. By correcting this single pre-analytical step, researchers could finally measure the total amount of PD-L1 present. This allowed them to reevaluate the raw data from the entire Taiwanese patient cohort. Returning to the 154 TNBC patients, we can now see the mathematical impact of the PNGase-F wash on clinical diagnosis rates. This chart tracks the standard, non-deglycosylated protocol at a tumor proportion score cutoff of 1%. The dark blue segment shows a baseline positive detection rate of roughly 73%. Based on the standard assay, approximately 33% of the cohort was classified as PD-L1 negative and theoretically excluded from immunotherapy. But when the exact same tissues were treated with the deglycosylation wash, the red column shows positivity jumping to nearly 98%. An astounding 84% of the cases initially classified as negative were reclassified as positive after the sugars were removed. The standard protocol was generating a nearly 28% false negative rate across the entire cohort. It systematically hid a subpopulation of patients who possessed the biological profile required for treatment. This raises a critical clinical question. Does detecting more PD-L1 actually matter if the tumor doesn't respond to the drug? To answer this, researchers isolated the subgroup of 74 TNBC patients who actually received neoadjuvant immune checkpoint blockade therapy. This graph compares the PD-L1 levels of patients who achieved a pathological complete response against those who did not. Look at the height difference in the red bars. The deglycosylated PD-L1 levels show a distinct, significant correlation with successful tumor eradication. Contrast spot with the blue bars for native PD-L1 scores, where overall expression stays low and fails to provide a clear separation between responders and non-responders. Exposing these hidden receptors provides a more accurate map of the PD-L1 density that correlates with the actual tumor response observed in the clinic. To quantify how much this enzymatic wash improves the assay's performance, we look to receiver operating characteristic analysis, or ROC, the standard statistical method for measuring a biomarker's predictive value. This ROC curve plots the standard native PD-L1 assessment using the blue line, yielding an area under the curve of 0.648. Tracing the red line reveals the deglycosylated assessment, jumping to an AUC of 0.860, a significant leap in predictive reliability. By clearing the N-glycosylation sites, the assay's ability to distinguish between responders and non-responders moves into a range associated with high-performing clinical biomarkers. The final metric for any oncology workflow change is whether it accurately predicts how long a patient will live. These Kaplan-Meier curves map overall survival using the standard native PD-L1 test. The lines blend together, yielding no significant association with survival. But when stratified using deglycosylated PD-L1 scores, the curves break apart, creating a clean divergence between high and low expression. Discovering the unmasked PD-L1 yields a significant divergence in survival probabilities, with a p-value of 0.028. These sugar chains create a gap between the physical presence of the target and our ability to detect it. Incorporating this enzymatic wash into the workflow identified an additional 28% of patients who possess the target density required for life-saving immunotherapy.